Thank you all for being here. Again, my name is Martinique Chavez. Today I'm going to be talking a little bit about the research I did for my master's, looking at the movement characteristics of the Rio Grande Silvery Minnow. So movement is a defining behavior in an animal's life. It has the capacity to connect populations, facilitate survival, and reproduction. However, insight into how, why, and when an animal moves is often insufficient. In particular, only 1% of riverine fish species have been considered in animal movement studies. So why do these movement patterns matter? Well, understanding the mechanisms, the potential drivers, and consequences of a species movement can be a valuable conservation status assessment tool. The quantification of a species movement can provide better insight into these larger ecological processes, which can be crucial during times of biodiversity loss and climate uncertainty. With recent advances in tracking and tagging technologies, we have the opportunity to gain insight into the movement patterns of these commercially unimportant small-bodied fish species. Like we just heard, the Rio Grande Silvery Minnow is a small-bodied and relatively short-lived species, and they are restricted to a highly modified and fragmented stretch of the Middle Rio Grande. So our study area encompassed the Angostura, Isleta, and San Acacia reaches. The goals of our research were to estimate the movement patterns of the silvery minnow and to quantify their movement metrics. We also aimed to estimate or develop a multi-state model that could estimate reach-specific transition probabilities. We used a mark-recite approach to characterize these movement patterns. So we used pit-tagged hatchery-reared silvery minnow and multiple antennas to track their movements. So we implanted the hatchery-reared silvery minnow with these passive integrated transponder tags, which are pit tags that provide a unique identification code for each individual. Now, given the small body size of the silvery minnow, we used the smallest available pit tag with sufficient scanning capacity. We were pleased to have a combined retention and survival rate to release of 88%. So between 2018 and 2022, a total of 37,215 fish were released at multiple locations spanning approximately 110 kilometers. So again, we used multiple antenna types to track these movements. These submersible antennas were kept in the river semi-permanently and detect tagged fish as they swim over. We also used these floating raft-mounted antennas. So we would launch 3 rafts simultaneously at the same location. We would make multiple passes with each raft covering a distinct section of the river. We conducted a total of 11 float trips and spanned 170 kilometers of the Middle Rio Grande. On to some of our results. We had an extremely high recite rate, detecting nearly 14,000 individuals out of the 37,215 released. In other mark recite studies, 20% is considered adequate, so this 36% is extremely high. So to account for that short lifespan of the silvery minnow, we only included individuals, or the detections from individuals that were within 365 days of a fish being released. And this also would reduce the effect of ghost tags, which are tags that are no longer associated with a live fish. We calculated the movement metrics for individuals that had at least 2 detections following their release. To account for the sinuosity of the river, we calculated this this total distance moved as the sum of the distances between detection points after release. So silvery minnow displayed a leptokurtic or heterogeneous distribution in the total distance moved metric. This means that a larger portion of individuals were not making long distance movements. They were remaining stationary and not moving very much. But a smaller portion were making these longer distance movements. In this slide, we have the frequency of tags detected on the Y and total distance moved on the X, with distances above 50 kilometers in that upper right-hand corner. So the maximum total distance moved by the silvery minnow was 103 kilometers. We also calculated this metric, this linear home range, and we did that as the difference between the upstreammost and downstreammost detection point over the course of 1 year. So the total distance moved by an individual could be larger than their home range size, but this metric, again, the difference between the upstream and downstreammost detection point, just describes the total 100% linear extent of the river that an individual used within that 1 year period. So the mean home range size for Silvery Minnow was 2.4 kilometers and the maximum was 78. In this slide, we have that linear home range size on the Y and the total distance moved on the X, with color representing whether the individual was ever detected immediately below the Santa Acacia Diversion Dam. So these clusters of points along the Y-axis correspond with detections that were made immediately below the dam, suggesting that it's acting as a barrier to the expansion of range sizes for many individuals. Overall, the total distances moved were larger than home range sizes for many of the fish in our study. We also wanted to assess any directional bias in these movement patterns, so we did this net directional movement metric, which was a net displacement, taking into account the directionality of the movements. So the distribution of these directional movements were left skewed or skewed downstream. In this slide, again, we have the frequency of tags detected on the Y and net distance on the X, with that vertical dashed line representing that first detection after release. 74% of the movements that we detected were directed downstream. Silvery minnow overall were highly mobile and displayed this ability and predilection to make long-distance targeted movements upstream to the extent possible and downstream. We documented silvery minnow moving farther than ever previously recorded. We also found little evidence of any upstream bias like that would be frequently ascribed to other pelagic broadcast spawning minnows. Silvery minnow generally displayed a nomadic movement pattern. So this movement pattern is often driven by environmental variation. Historically, silvery minnow persisted under extreme environmental variability, were often subjected to these long periods of drought. So this historical persistence likely required access to refugia during these periods of river drying and subsequent connectivity to recolonize upstream reaches. So these patterns, combined with the long distance movements and the high reproductive effort of the silvery minnow and their short lifespan, suggest that they would be able to recolonize upstream reaches over relatively short periods of time with sufficient connectivity. So our next goal was to develop this multi-state model that would estimate the transition probabilities between reaches. So our model was focused on accurately estimating upstream and downstream movements across spatial states. The spatial states in our model were delineated by the Santa Acacia Diversion Dam. So state A was defined as everything above the dam, state B was a 1 kilometer stretch immediately below the dam, and C was everything below state B. We defined the time steps in our model using these 4-month intervals that correspond with the irrigation system in the Middle Rio Grande. So we only included, constructed encounter histories for individuals that had at least one detection in a subsequent time step after their release. So for these individuals, we separated them into 2 cohorts depending on the season that they were released. We then assigned a group covariate to these cohorts depicting whether or not they were released above or below the dam. So out of these individuals, 63% were released below the dam. So we used a Bayesian hierarchical approach with a Markov chain Monte Carlo estimation in program MARC to estimate these transition probabilities for the silvery minnow between spatial states. This table shows the actual state transitions. We can see that 61 to 67% of these fish remained in the same state. In this slide, we have the transition probabilities for our winter release cohort. And on the, let's see, the right-hand side here, we have the individuals that were released below the dam, and on the left, those released above. And the width of these arrows correspond with the transition probability. So we look at state C, you can see that there's a higher probability of those individuals remaining in that state, especially compared to state B and state A. We also see the highest upstream movement probabilities in this winter release cohort. When we look at the spring release cohort, we see much lower probabilities of upstream movement and even higher probabilities of remaining in the same state. So just to clarify, these transition probabilities are for individuals that, say if you're released in the spring, this probability will be in the summer time step, so that's in the subsequent time step. So this multi-state model provides pretty crucial insights into the spatial dynamics of hatchery-reared fish after their release. Overall, we saw higher probabilities of upstream movements for individuals that were released in the winter. Also, transition probabilities were highest in that first time step. So if an individual did not move in that first time step, they were less likely to move in subsequent time steps. About a third of individuals that were released above the dam remained above the dam, and 2/3 went below. The majority of those individuals that were released below just remained below the dam. So the Santa Acacia Diversion Dam is not equipped with a formal fish passage structure, which is why we were surprised to document a total of 198 unique upstream passages and 2,052 downstream passages. So the dam gates at Santa Acacia can be open or closed for water management, but regardless of the gate position, the route where we believe that passage is occurring has extremely high velocities. So these individuals that successfully made it upstream were doing so under very narrow circumstances and displaying an intent and determination to expand upstream. When we look at these upstream dam passages by season, we see that the majority occurred in the early irrigation season between March and June. In this slide, we have the number of upstream dam passages on the Y and season on the X, with color representing season at a finer scale. So although these upstream passages are impressive, just as our multi-state model predicted, they only account for less than 1% of the total fish that we included in our analysis. Studies like ours address the lack of small-bodied fish movement studies. Our approach embraces this emerging tracking and tagging technology, and it advances our ability to understand movement patterns in fragmented systems. This knowledge can help define appropriate scales for management and monitoring efforts. The direct evidence of the long-distance movements that we saw in the Silvery Minnow, as well as those upstream dam passages, and the repeated number of detections immediately below the dam suggest that ranges could be expanded with sufficient connectivity. All of this highlights the need to restore connectivity through either the removal or redesign of diversion dams to include fishways. Lastly, given the widespread fragmentation of desert rivers, our research serves as an important model that can be applied to other small-bodied fishes in fragmented systems. Our multi-state model can be expanded upon and replicated to assess the efficiency of fish passage following dam modification, particularly in these small-bodied fishes. Overall, a better understanding of how fragmentation impacts the movement patterns of these imperiled fish species can help guide and better inform management and recovery efforts to ultimately achieve self-sustaining populations of native fishes. I'd like to thank my co-authors, Dr. Phaedra Booty, Dr. Mary Connor, and Dr. Casey Pennick, as well as our funders at the Bureau of Reclamation, Utah State University, U.S. Fish and Wildlife, in particular Thomas Archdeacon, American Southwest Ichthyological Researchers, in particular Robert Dudley, the USGS Utah Cooperative Fish and Wildlife Research Unit in kind, and with that, I will take questions.
Great. Great, thanks, Martinique. That was really great. That was a really interesting talk. It made me think of a lot of questions too. But I'll take questions from the audience.
Martinique, nice talk. So I got a couple questions for you. How did you account for the ghost tags? I mean, is it explicitly accounted for in your model?
No, and that's kind of the nice thing about working with the silvery minnow that a lot of you run into with these long-lived species. Because they are short-lived, we were able to just subset our detections to that 1-year period. It's not going to completely account for them, but it's going to reduce the possibility of having those ghost tags. But we did have multiple passes with our float trips to ensure that the fish that we were tagging were live. So we'd do 3 days in a row the same stretch of river, and we can kind of parse out certain tags depending on if they were moving with the sediment or if they were, just to say if they were shed, if they were lost by a tag or it wasn't a biologically feasible thing for them to be alive.
Gotcha. And then the second question is a much quicker one. So do you have, are all the tag sites, the detection sites georeferenced? Mm-hmm. And precisely georeferenced?
Yes.
Okay, excellent. Thanks.
Thanks. We have time for another question. Is there any more questions? Very good.
So it looked like, if I was reading that chart correctly with the dam and the ABC and the circles and how they moved, it looked like in the springtime that upstream they were staying upstream. Is that what you found, and is that consistent with your kind of thinking about it?
Yes, so in that winter release cohort, those fish, I think it was about 40%, if I remember correctly, that moved upstream to a different state remained in that state in the following time step.
I had a question, if you don't mind. I'm learning a little bit more about fish, but from my experience with birds and mammals, when you do a release, when you reintroduce an animal, the first sort of instinct is to just move, right? And to just get out of there or to explore the area widely. So what I'm wondering is, how reflective do you think some of these measurements are of how their movement distances are of the natural population?
Right, and so that's something that is pretty well documented in hatchery fish, is they have this tendency to move downstream immediately after being released. And so what we did was we subset our detections, like we got rid of the first week, the first month after they were released, and it didn't change the output of any of the analysis that we ran. And also I'll just point out that there's some literature to support the fact that after 6 months, if a hatchery fish, still alive, that they're essentially functioning as a part of the wild population.